So next is Warispreet Singh. Here he is. Waris's talk today is about rational engineering of enzymes for improved activity and selectivity using computational approaches. The floor
Hi, Gary.
is yours.
Can you
yes,
see me?
I can say that and I can hear you.
So you can see my screen, yeah?
Yep,
okay.
All good.
Yeah, thank you very much for giving me this opportunity to present my work. So I will talk about rational engineering of enzymes for improved activity and selectivity using computational approaches. So in this short presentation, I will go through why we care about computer simulations. Second, what is directed evolution of enzymes, and how these computer simulations can help us in the directed evolution of enzymes. So why do we need simulations? One of the very famous scientists, Richard Feynman, once said, everything that living things can do could be understood in terms of jiggling and wiggling of atoms. And from the past few decades, we know now molecules are dynamic. They're not static. So here I've shown a very iconic molecule in chemistry. It's a ruthenium bipyridine complex. This is a supramolecular chemistry concept of host-gas interactions. And here you can see how the host, which is gray, is accommodating the gas, which is ruthenium bipyridine complex. So here we show on the top hand side the reduced form of the host. When it goes to its oxidized form, you can see it get rid of the host, get rid of the gas molecule. And this actually matches very well with the spectroscopic data. So in essence, if we do computer simulation, we can have in-depth detail of what is going on, how these two molecules interact. We can compute all sort of properties. We can compute the various electronic states of this molecule. We can compare this to the spectroscopic data. So now I would very briefly go through what directed evolution is, and as you all know, it's a multidisciplinary approach. So, for example, if you have an enzyme, a wild-type enzyme, and you're interested in a certain chemical transformation, but you want to improve that chemical transformation, what you can do is you can add mutagenesis to the wild-type enzyme using molecular biology techniques. You can build those mutants, you can screen the mutants, and then you can learn from the experiment. And by doing many rounds of this directed evolution, you fine-tune the function, And in terms of its applications, people have used it to improve the catalytic efficiency of enzymes, to broaden their substrate scope. Industry have used that to obtain stereoselective products. You can improve the thermostability of the enzymes, and also some of the applications could be to use to make enzyme resistant or to work in the organic solvent. And this success of this directed evolution was awarded to the pioneer work of Professor Frances Arnold in terms of her winning the Nobel Prize. So it is now a mature field. This slide talks mainly about the computational perspective on the directed evolution. So in the past, directed evolution was mainly focused on experimental part, which is random mutagenesis and other molecular biology techniques. However, this process required a lot of mutants and required a lot of time and money. As we progressed further and our knowledge of the structures improved in terms of, by techniques such as crystallography, NMR, cryo-EM now these days. So we entered the realm of semi-rational based design, which was mainly focused on the structural information. And then now what we are doing a rational directed evolution, which is mostly simulation X-ray based design. And this is what I do mainly in the HPBE. So we perform in our In my group, we perform molecular dynamic simulations of enzyme substrate complexes to see how they interact with each other. We perform free energy calculations in terms of how the particular ligand would bind with the receptor, what is the penalty energy cost if you mutate one residue with another. Excuse me. And finally, we study the overall reaction mechanism, the catalytic mechanism, using quantum mechanics and spectroscopy. The last part in this graph is the artificial intelligence, which I think are very, very rapidly proceeding towards artificial intelligence, and how we can use the neural networks to help us design different mutations. But end of the day, still the bottleneck in this directed evolution is the experimental approach. So by using rational based design or artificial intelligence, we can narrow down the mutation scope. So we have to screen less mutants, and that will save us time and efforts. So just a few quick examples of what we have used, how we have used this rational based design in terms of directed evolution of CALB. And enzyme, it's a lipase, it's very important in industry. You can see the wild type active site consists of a textbook catalytic triad. Is actually quite similar to the protease enzyme, which Professor John talked about this morning. And we were quite interested to see, can we make this enzyme, the turnover more, make this enzyme more faster? And also, can we improve the substrate scope so that it can accept a bulkier substrate? And by using the approaches which I spoke to you in the last slide, we were able to make an artificial lipase, which was 40-fold, has 40-fold improvement in catalytic efficiency. And it was able to accept the bulkier substrate. So we were able to publish this. So this is obviously quite a multidisciplinary work where we did the computation work and Professor Wheat's group did the experimental work. We also applied the similar approach to obtain quite, very stereoselective isomers, again with CALB, and this was also published in Journal of American Chemical Society. In the context of HBBE, as Professor Gary mentioned this morning, we are very keen to work with the lignin breakdown, and particularly we are focused on GCOA enzyme. Lignin, as we know, it's a heterogeneous aromatic biopolymer. It's a very abundant source of aromatic carbon. It is mainly composed of 3 subunits, which I would pronounce as G, S, and H unit. However, to have the efficient depolymerization of lignin, we need to remove this methyl group. And indeed, aryl O demethylation is the reaction which was catalyzed by GCOA enzyme on this G unit to perform this catechol product and formaldehyde. The paper appeared in Nature Communications. So our idea was to, the crystal structure authors described, even though the wild type was able to accept this G unit, but they didn't have much activity with the S unit. So what they did was they, I believe they used rational design as well to pick this phenylalanine 169. And then they mutated this to alanine. And by doing that mutation, they were able to catalyze this S unit, which is, very fantastic. However, still there was some missing links, because in the experimental paper, which was published in Proceedings, they used this mutant to catalyze the S unit through 3-MC intermediate. However, the fate of this intermediate was not very clear, how this intermediate was converted to pharyngeal. And also, because these are monooxygenase, what means is they can only insert one oxygen atom into the organic molecule. So how did this enzyme regenerate to accept 3MC as the substrate to perform this catalysis? So these are a few questions which we are addressing using MD simulations and free energy calculations. However, in the context of HPB, we are also very interested to further improve the turnover of both S and G units and make this enzyme able to accept bulkier substrates. So we came up, so this is the active site. These results are quite similar to the papers published in Proceedings and Nature Communication, but we want to focus on these residues, which I've labeled, which I'm showing with my cursor, and to see if we can mutate them and we can increase the turnover for S and G units and make this enzyme accept bulkier substrates. So just in conclusion, we can use a structure-guided rational design to accelerate directed evolution. By doing that, we can improve the catalytic activity, we can improve the substrate scope, make enzymes more thermostable, and finally, we can, obtain novel biocatalysts for HPB. I would like to thank you, HPB, for giving me this opportunity. I'd like to thank especially all the people at Queen's University Belfast and here at Northumbria University who helped me throughout, and the high performance computing facilities provided by Queen's University, by Northumbria University, and our GPU cluster provided by HPB. And I thank you for giving me time, and I'm happy to take a few questions. Thank you.
Thank you very much, Waris. Really good talk, really interesting. Yeah, I had I think it's time probably for one question. I had a quite general question. Have you ever seen situations when the rational design that you perform doesn't pan out as far as the experimental work is concerned? And if you do see that, is there something that where rational design just doesn't simply work? Because I know there was a paper fairly recently when essentially a lot of crystallographers were getting very worried because it's sort of suggesting that maybe there's no need for crystallography eventually because we'll just be able to do everything via computational analysis. But
yes,
do you still see some things that can't be answered and via the rational design work?
Oh yes, as I say, I shouldn't be saying this, but end of the day, in terms of science, experiment decide if it's right or wrong. End of the, we work with the computational models. There are a lot of approximations. Sometimes we get sometimes we get the answers wrong. Well, most of the time we will get the answers wrong. But sometimes, it could be due to the different approximations used in the model, and also we don't understand enzymes that great till date, So that is, that. But yes, we fail quite a lot of time. But when things work, they work very beautifully, as we demonstrated in our work with lipase B, where you can obtain very highly selective stereoisomers, which is very difficult to obtain using traditional catalysts.
Yeah. Okay. So there's still jobs for lab-based scientists. That's
100%. I think chemistry relies on experiment. So I think we need experimental people, to test things. Yes, absolutely.
Brilliant. Well, thanks again for that. We'll move on to our next speaker, and our next speaker is Rajesh Bomareddy. Rajesh's talk today is sustainable production of fuels and chemicals from waste carbon. The floor is yours, Rajesh.
Can you hear me, Gary?
I can hear you.
And can you see this?
Yeah, it's not the slide format yet, though. We can see things up the side.
Is that good?
Still not full screen.
I think that might be Waris's we're seeing.
Are we still? Waris, have you there we go.
Oh, yeah.
Well spotted, Paul. Thank you. Over to you,
Yeah. You can hear and see my screen, right?
Yes, all good, all good.
Yeah. Thanks, Gary. So I'm Rajesh Vomareddy. I'm a VC fellow at Northumbria University, also working in the hub in the team building metabolism. And I'm working in this very interesting group, which has very different areas of expertise, and I would like to combine all this expertise in producing sustainable fuels and chemicals from waste carbon. So within this context, waste carbon is possibly we can say that it's mainly coming from the household waste, for example. So I'm by background, I'm an industrial biotech, so I don't have to really define what is industrial biotech now after Liz's talk. So I think that's clear now, and I have been working on strain development, fermentation process. So I try to always combine these two particular areas, which are quite important for creating this carbon and energy efficient circular economies, which are quite important now in a way of transformation from a fossil resources towards a biomass feedstocks, for example, in this context, waste. My vision is mainly, I would like to work in a very integrated manner to create this particular sustainable platforms to produce energy rich compounds, like it could be chemicals, fuels, even low value, high volume chemicals or fuels, or even interested in producing high value, low volume pharma products, for example. And if, as you see here, this particular overview of I call this my biomanufacturing process, which is an integrated process. And you can see that as shown by Liz as well, that we need to have a efficient pre-treatment technique. Of course, we have biological pre-treatment techniques like enzymatic pre-treatment techniques, as well as chemical thermochemical pre-treatment techniques to basically break down this complex waste feedstocks, including plastics, waste plastics, for example. All this has to be broken down. So one of the biggest problems with microbial conversion process is that they really like some very basic building blocks like sugars, carbon dioxide, and things like that. So the idea is to break them efficiently. So I say efficient because these are is one of the limiting steps in industrial biotech sector. So once we have a feedstock chosen, and then we could efficiently break them down using chemical or enzymatic pretreatment techniques. Currently, I would say the enzymatic pretreatment techniques are still being developed. They are under kind of research. I would even call certain thermochemical processes like gasification process are being widely used to break down these molecules and to produce energy. So these are certain examples, but all these techniques individually, I think they are not very efficient as of yet. So once we break these complex structures into these very basic components, as Paul was also mentioning, this is a part of synthetic biology that he was mentioning. And I'm talking about a wider perspective here of a systems metabolic engineering cycle, which is a similar cycle like design, build, test cycle. And we'll be working with certain in-depth experts here like Waris, who can do the synthetic biology aspects of enzyme engineering. And then we have Paul, who will be doing synthetic biology as well, constructing and creating new designs. And then we also have a systems biology, so I can do systems biology as well. And cycle runs, as said, the iteration runs until we create that particular potential strain, which shows this potential TYP metrics, I call them, which are tighter yield and productivities from the given substrate. And once we have that particular strain, then we demonstrate it in a fermentation intensification process, an optimal process where we can produce efficient amounts of this product from the given substrates. And of course, we do a techno-economic analysis to identify if our process is anywhere near to commercialization. We also do a life cycle analysis to see if the greenhouse gas emissions from this process is negative. And then finally, we have really a green product. And I would like to stress that to make this process very carbon and energy efficient, it only works when everything are integrated in a manner, because there are certain processes which are energy intense, and there are certain processes which are energy producing processes. So if there is an integration between both of them, there is an energy saving step. So that makes the process a complete efficient process in producing any bioproduct. So I just want to go into some case studies. I would like call them examples. So I'm mainly interested in breaking down these molecules currently to smaller molecules like CO2 and hydrogen. So I'm quite interested in an emerging technology called supercritical water-based gasification. It has certain advantages over the traditional gasification. It can produce a lot of hydrogen, which I said, when we integrate this particular process, we can reuse this hydrogen to basically generate energy for the process itself. So that makes it an interesting and a very nice technology in an industrial biotech perspective in treating these complex structures. So I would like to use that technique, and we can break down any complex structure. There were studies demonstrated on breaking down waste plastics using that technology and producing hydrogen from it. And also this supercritical water gasification was also used to break down household waste, municipal solid waste, producing hydrogen from it. So my main interest is using that particular technology, break it down, break any complex structure to CO2 and hydrogen, and then use that in a microbial conversion process using, for example, in this case, cuprividis nicator. So the organism is a chemolith autotroph. So as opposed to Shafir's photosynthesis, this is also running a metabolic pathway that we see in photosynthesis called Calvin cycle. But this uses hydrogen as energy donor. So this kind of the process works in that it uses carbon dioxide as a sole carbon source, hydrogen as an energy electron donor, and oxygen as electron acceptor. And using this particular organism, we have demonstrated at scales like really gram per liter scales. We're not talking anymore about, especially with cyanobacteria or this kind of organisms, we always talk about milligrams. But in this case, I'm talking about really gram per liter scales. We're also working on a biofuel production in a sustainable aviation fuel, which we use isobutanol as a precursor for this, where my PhD student Toby is working on it. And we have certain other precursors which can be converted to certain very important products, like for example mevalonate can be converted to polymers. Butanediol has a high value in pharma products. Isopropanol can be used as a blend in fuels, and even R2,3-butanediol. So we all were demonstrating well, and this organism is proving to be very efficient in carbon conversion efficiencies comparatively to any photosynthetic kind of organisms, mainly fixing CO2. So to make this process work efficiently, we need a setup. We need a gas fermentation setup, because I was talking about hydrogen combined with oxygen. So this is a very potential toxic or flammable atmosphere. Could be, we have safety risks with this. So at previous workplace, we designed a system where we can work efficiently with this organism using hydrogen and oxygen, and this particular system is used. I don't want to go into detail in the system, and it kind of helped us. Basically, it's a process optimization step that we developed at Nottingham to produce this kind of gram per liter scales. And I'm saying efficiency, so as you can see, it's a case study where isopropanol was produced together with acetone as a product, as a byproduct in this case. And you can see the conversion efficiency of CO2 is 80%. So there's almost like 80% of carbon of CO2 is converted to the product. So the carbon conversion efficiency is quite high, but the amount of hydrogen that is required per carbon dioxide is quite high. So you can see here for the two products, 2,3-butanediol and isopropanol, the amount of hydrogen required per CO2 is like about 7 to 8 moles. So it's as seen, Calvin cycle is a very high energy intense metabolic pathway. So it kind of needs this high amount of hydrogen, which is the main cost driving force for this particular gas fermentation process. So we looked into areas where we can improve this efficiency, that is reducing the hydrogen requirement per carbon dioxide. On genetic basis, I did some simulations in a pathway analysis, and I identified certain targets which can reduce the hydrogen per CO2 molecule used for the particular products produced. So that's under publication is considered. And we also dealt with dealing with this efficiency based on process. So what can we do? As I was talking about integrated process, can we solve this within process optimization? And we have designed this particular process flow of heat integration. As I said, combining the heat from the gas fermentation and delivering it to the supercritical water-based gasification using a heat pump, we could actually achieve a process which is sustainable on its own based on the hydrogen that is produced. And we demonstrated using this particular process. We did a techno-economic analysis for isopropanol being produced from waste carbon, from black liquor in this case. And we can actually see that we projected a positive net present value for isopropanol. But of course, it has to be, the cost of the product is the point here. So it has to be costed at $100 a ton. Then we can have a positive net present value for that product. And also comparing it with the already present processes like LanzaTech's gas fermentation producing ethanol, our isopropanol production plant also has a favorable greenhouse gas emissions. So it has a negative greenhouse gas emissions, which is positive, and it also has favorable NPV, net present value. So it's a very important step in determining if aerobic gas fermentation is a viable process, if it is a techno-economically viable process. We also are working on a proof of concept grant where we propose designing a bioreactor, a different way of working with this autotrophs which use hydrogen. So the idea is to use a microbial fuel cell in this case to separate oxygen and hydrogen, basically, and then use microorganisms on the anodic side, donating electrons to the anode, and then we produce electricity in this case and water on the cathodic side. And the organism can be engineered to produce any product. For example, in this case, we are demonstrating it using butanediol being produced. So concluding from this, as said, waste offer a very important like outlook for us to create circular economies. They are in abundant renewable feedstocks available, but we do need efficient treatment techniques for this, especially on the biological side. We need a very efficient. We are still under progress in developing very efficient treatment methods. And definitely, as shown, we need an integrated technologies. So to create this efficient, carbon efficient or energy efficient technologies, we need to integrate them. And then this would drive us towards an efficient and sustainable biomanufacturing of products. Thank you.
Thank you, Rajesh. Really good talk. I think there's probably time for one question. Could you give us a feel for the amount of energy you have to put into the system to perform supercritical water gasification? Obviously, I know you've done some life cycle analysis, and actually, you know, the amount of energy you get out is presumably more than what you put in. But is it a significant amount of energy that has to go in to perform supercritical water gasification?
Yeah, it is. I mean, I don't know how to measure it. We have numbers, but I don't think that will give you a good idea. But if you consider a normal incineration, like a gasification, normal one, not the supercritical one.
Yeah.
This one uses half or 3/4 of what is required, because if you combine that for gasification, you need to dry the biomass and then implement huge amount of energy. But for this one, you don't have to dry. It works with at least 30% of moisture content. So you just take almost waste carbon, for example. This was demonstrated using black liquor from waste from paper and pulp. So there is significant less. There are certain advantages of it, and I would say there are some disadvantages. You know, some ash formation and things like that. But it is certainly a developing technology. So all this were dealt with.
Brilliant, excellent. Okay, we'll try to keep to time, so we'll move on to the next speaker. But thank you very much for that, Rajesh. Really interesting talk.